Now I am showing PowerPoint presentation for the journal article Nanotechnology in Respiratory Medicine. Over the past few years, much effort is done to adapt nanotechnology for the treatment of human diseases. The term nano refers to structures in the range of 1 to 100 nanometer, in contrast to nanoparticles, which have to measure between 1 and 100 nanometer in all dimensions. Nanomaterials may consist of elements bigger than 100 nanometer but need to be structured in the nanoscale and exhibit characteristic features associated with the nanostructure. For this, the International Organization for Standardization defined the term nanoobject as a material with one, two, or three external dimensions in the nanoscale. Nanoobjects can be divided into nanoparticles, nanofibers, and nanoplates, depending on their number of external dimensions in the nanoscale. Nanoparticles have three external dimensions in the nanoscale. Nanofiber has two external dimensions in the nanoscale, and nanoplate has one external dimensions in the nanoscale. Nanomaterials have an extremely high surface area to volume ratio. Therefore, some of them are very reactive or catalytically active. Like viruses and cellular structures, some nanoparticles are able to self-assemble to more complex structures. This makes them interesting candidates for the novel drugs. It is also necessary to redefine toxicology because of nanotechnology. Unlike classical toxicology, where dose and composition matter, in nanotoxicology, the focus has to be set on properties like morphology, size distribution, surface charge, and agglomeration state as well. Nanotechnology offers new approaches to treat diseases of the respiratory tract. The use of nanotechnology in consumer products, cosmetics, and medicine is continuously increasing. Therefore, it is also pivotal to understand potentially adverse effects of nanomaterials on the respiratory system. Additionally, the study of respiratory effects of manufactured nanomaterials helps to understand the impact of combustion exhaust and ultrafine dust on human health. On top of that, the lung is probably the most important gateway of nanoparticles to human organism. For assessment of safety in nanotechnology, it is therefore also important to elucidate which nanoparticle properties determine pulmonary resorption and biodistribution. Applications of nanotechnology in therapeutics and diagnostics. The nanoparticles are in increasing use for respiratory medicine in three main areas. First of all, clinical use of nanotechnology. We will study drug delivery, hyperthermia, gene therapy, and contrast agents. Secondly, the use of nanomaterials can cause toxic effects in the respiratory system, which includes immune effects, oxidative stress, and genotoxicity. It is also necessary to learn the pulmonary absorption and distribution to explore extrapulmonary organs and the effects of inhaled nanoparticles. Clinical use of nanotechnology. A. Drug delivery. Nanoparticles can act as vessels for drug because they are small enough to reach almost any region of the human organism. Drugs can be bound chemically to the nanoparticles by a multitude of different linker molecules or by encapsulation. The main advantage is the capability of the targeted drug delivery. The targeting can be active or passive. In case of tumor diseases, the leaky and immature vasculature of fast-growing tumors can be taken advantage of in order to achieve passive targeting of chemotherapeutic loaded nanoparticles. This is called the enhanced permeability and retention effect. The first generation nano drug delivery systems rely entirely on the EPR effect. One example is Xenoxol PM, a polymeric paclitaxel loaded missile formulation. This nanocarrier has recently been tested in a phase 2 trial in patients with advanced non-small cell lung cancer. The second generation nanoparticle drug delivery systems possess targeting ligands. These can be antibodies, aptamers, small molecules, and proteins. The attached ligands actively guide the nanoparticles and therefore the drugs to the tumor cells. Tumor-specific monoclonal antibodies are already widely used in cancer therapy. Those antibodies can be attached to nanoparticles for active targeting. Another approach involves aptamers as targeting agents. Aptamers are synthetic oligonucleotides that are capable of binding specific target structures. Their small size, their simple synthesis, and their lack of immunogenicity make them promising ligands for nanoparticles. Nanoparticle-based drug delivery system also offers potential in other fields of respiratory medicine. In experiments with tuberculosis-infected guinea pigs, it was demonstrated that inhaled alginate nanoparticles encapsulating isoniazid, rifampicin, and pyrazinamide showed better bioavailability and higher efficiency than oral drug medications. Next is hyperthermia. Nanoparticle-induced hyperthermia can be used to locally destroy tumor cells. Heat generation is usually achieved by two approaches, magnetic and photothermal hyperthermia. Photothermal therapy uses laser radiation in the visible or near-infrared spectrum and photosensitizing nanoparticles such as gold or graphene. A commercial product called Oroshel is available for tumor therapy. Oroshel nanoparticles consist of a silica core surrounded by a thin layer of gold. A gold nanoshells are administered intravenously and accumulate in the tumor due to the EPR effect. Upon exposure of the tumor to a near-infrared laser, the laser energy is efficiently converted to heat by the gold nanoshells. This therapy, which is called Orolase, is currently undergoing clinical trials in patients with primary and/or metastatic lung tumors. Third is gene therapy. Like viruses, nanoparticles can be used as vectors for gene, but in contrast to viruses, they are less immunogenic and have higher DNA transport capacity. In a study, DNA-loaded polyethylene nanoparticles were used to treat lipopolysaccharide-induced acute lung injury in mice. After IV injections of the nanoparticles, the beta-2 adrenic receptor genes in the nanoparticle led to a short-lived transgene expression in the alveolar epithelial cells. As a result, the 5-day survival rate is improved from 28% to 64%. DNA-loaded nanoparticles are also promising candidates in treatment of cystic fibrosis. It was shown in a clinical trial that nasal application of DNA nanoparticles is safe and evidently leads to vector gene transfer. One major problem in this context is to overcome the mucous barrier. A recent study showed that densely PEG-coated DNA nanoparticles can rapidly penetrate extracorporeal human cystic fibrosis and extracorporeal mouse airway mucus. Those particles also exhibited better gene transfer after intranasal administration to mice than conventional carriers. Nanotechnology is also used in diagnostics. Nanoparticles have potential to improve pulmonary X-ray diagnostics. Folic acid modified dendrimer-trapped gold nanoparticles were utilized as imaging probes for targeted CT imaging. Nanoparticles can also be used to enhance hemodiagnostic of lung tissue. In experiments with intratracheal administration of gadolinium DOTA nanoparticles in mice, signal enhancements in several organs, including the lung, were measured with ultra-short echo time for proton MRI. The signal change over time demonstrated the passage of nanoparticles from the lung to the blood, then to the kidneys and finally to the bladder. It is also important to discuss about toxicological aspects of nanotechnology. Ultrafine particles of low soluble, low toxic materials like titanium dioxide, carbon black, and polystyrene are more toxic and inflammatory than the fine particles of the same material. To correlate the toxic effects with nanoparticle properties, it is necessary to thoroughly characterize the selected nanoparticles prior to administration. Most commonly used methods to characterize the nanoparticles for toxicology studies. First is transmission electron microscopy for size, morphology, and agglomeration. Second is dynamic light scattering for size distribution of the particles, zeta potential measurement for nanoparticle surface change. The third is X-ray diffraction for particles crystal structure. The toxic aspects of the nanotechnology can be studied in three parts. First is effects on immune system and inflammation. Second is oxidative stress, and the third one is genotoxicity. First, effects on immune system and inflammation. Many nanoparticles possesses properties that give them the potential to influence the immune system, such as ability to penetrate cellular boundaries, to escape the phagocytation by macrophages, to act as heptans, and even to disturb the Th1 Th2 balance. For carbon black nanoparticles, a recent study investigated the effects of inhalative exposure on mice with bleomycin-induced pulmonary fibrosis. The analysis of histology and cytokines expression suggested that the nanoparticles triggered an inhalation exacerbated lung inflammation. The author concluded that, especially for people with pulmonary preconditions, inhalation of nanoparticles can lead to serious health problems. Some nanomaterials have impressive immune modulating activity. As an example, gadolinium compound, a fluorine derivative with a gadolinium atom inside, showed anti-cancer activity without being cytotoxic. OVA sensitized mice that were additionally treated with the nanomaterial graphene oxide during allergen sensitization had stronger airway remodeling and hyperresponsiveness than mice that have only been treated with OVA. The graphene oxide led to a downregulation of Th2-dependent markers such as IL-4, IL-5, IL-13, IgE, and IgG1, but it increases Th1-associated IgG2A. This kind of immune modulation can also be utilized for therapeutic purposes. In a recent study, a nanoparticle-based vaccine has been used to treat dust mite allergies in mice. Mice treated with those nanoparticles showed significantly lower airway hyperresponsiveness as well as lower IgE antibody levels after a 10-day intranasal DERP2 instillation compared to the control group. This biodegradable nanoparticle-based vaccination strategy has significant potential for treating HDM allergies. Oxidative stress. Oxidative stress is measured directly with dichlorofluorescence or indirectly by the upregulation of reactive oxygen species, eliminating enzyme like superoxide dismutase. Another approach involves test whether the nanoparticle dependent toxicity can be reduced by the application of an antioxidant. Widely used semiconductor materials, such as lead sulfide nanoparticles, may have potential to generate oxidative stress in the lung. A recent study tested the toxicity of intratracheally applied 30 nanometer and 60 nanometer lead sulfide nanoparticles on the rats. Oxidative damage was evaluated based on superoxide dismutase, total antioxidant capacity, and concentration of malondialdehyde. In addition to inflammatory responses, both 30 nanometer and 60 nanometer groups showed increased oxidative damage compared to control groups. Genotoxicity. Genotoxin is a chemical or agent that can cause DNA or chromosomal damage. A common method to quantify genotoxicity is the COMET assay, which uses electrophoresis to detect DNA strand breaks. This assay was used in a recent study to check whether intratracheal instilled fluorescent C16 nanoparticles induced DNA damage in the male rats. However, there was no significant increase in fractured DNA in the lung cells. Therefore, it was concluded that even at inflammation-inducing doses, fluorescent C60 nanoparticles have no potential for DNA damage in the lung cells of rats. Another study demonstrated that intratracheal instillation of anatase titanium dioxide nanoparticles on rats did not result in genotoxicity. None of the titanium dioxide groups showed an increase in fractured DNA, while the positive control with ethyl methanosulfonate exhibited significant increases. In contrast to those results, even in low doses where no inflammation occurs, Brindex 90 carbon black nanoparticles induced genotoxicity in mice. There was no inflammation, cell damage, and acute phase responses, which means that the increased DNA strand breaks are related to direct DNA damage caused by nanoparticles. On the other hand, a recent study suggests that CEO2 nanoparticles may be even used as antioxidant and anti-genotoxic agents in the lung. After treatment with the oxidative stress-inducing agent KBrO3, VEAS2B cells pre-treated with CEO2 nanoparticles showed significantly less intracellular ROS as well as a reduction in DNA damage compared to non-pre-treated cells. Third is biodistribution, nanoparticle detection. The biodistribution of nanoparticles requires tracking of applied nanoparticles in the test animal. The conventional light microscopy is not able to detect nanoparticles. Electron microscopic imaging is often required to detect nanoparticles. However, light microscopy can be used to describe the nanoparticle-induced changes in the cell morphology without being able to see the nanoparticles themselves. Nanoparticles can be indirectly made detectable in the light microscopy by a method called autometallography. This is a silver staining that can be used to increase the size of several types of nanoparticles like gold, silver, and some metal sulfides and selenides in the histological section. Particle deposition and resorption in the respiratory tract. Most research about biodistribution of nanoparticles in organisms focuses on intravenous injection. However, nanoparticles were shown to be able to pass the blood-air barrier of the lung. Nanoparticles passing through the lung into the body seems to be size-dependent. This was evaluated by injecting neutron-activated radioactive gold nanoparticles of 1.4 nanometer and 18 nanometer intratracheally to rats. The bigger nanoparticles almost completely retained in the lung, while the significant amounts of the smaller 1.4 nanometer particles were found in blood, liver, skin, and carcass 24 hours after installation. In addition to physical parameters of the applied nanoparticles, the health status of the exposed organism also seems to play an important role. Conclusion: over the last decade, major breakthroughs in nanotechnology have been achieved. It is only a matter of time before new nano-based drugs reach respiratory medicine. The fields of targeted drug delivery, gene therapy, and hyperthermia offer great potential for modern drugs. On the other hand, increased use of nanomaterials in all fields of life also bears the risk of exposure through inhalation. It is therefore essential to understand pulmonary toxicology of nanomaterials in all its facets. However, the toxic effects of nanoparticles in the respiratory tract are so inhomogeneous and not well predictable. In this context, not only local reactions of lung and airways, but also nanoparticle uptake and distribution in the organism are important factors and therefore should be researched further. As only few nanoparticle compositions have been tested, it is questionable whether those results can be easily adapted to other nanoparticles. Because of continuously increasing diversity of engineered nanoparticles, toxicology can hardly keep pace with safety assessment of future products. Therefore, more attention should be set on this wide field of research. Thank you.